In this video, we'll discuss some techniques for finding and validating ideas for biotech startups. So the previous video described the overall process that we use for developing and validating hypotheses. Basically, you come up with several ideas, triage them to identify the idea that is initially the most promising, and then subject that idea to negative feedback. Most ideas won't survive that scrutiny, which is fine. The goal is to keep iterating until you find an idea that does stand up to all the negative feedback you can throw at it. So this video is just going to talk about the first steps of that process. How do you come up with ideas, and then how do you pick which ones to focus on first? So the first step is to brainstorm venture hypotheses. So you may remember this slide from the last video, but this is just basically intended to be a reminder of what a venture hypothesis is. So basically, you want to come up with hypotheses of the form: a new product based on these specific technologies can solve this problem for this set of patients. And then through solving this problem, you could create a billion-dollar company, which is what you'd need to do to convince investors to fund you. So these are just some examples. We talked about them a little bit last video. But the important thing that we're talking about now is how do you generate these ideas? So we'll go through a couple ways you could do so, but there's really countless ways to come up with ideas. There's no right way, there's no wrong way. It could be different for everyone. And you may have a ton of ideas already, or be able to come up pretty easily with a lot of interesting ideas. But if you're having trouble, then we'll talk about a few frameworks to use, and then we'll also talk about a couple frameworks for evaluating which ideas are promising or not at first. All right, so one of the most common ways to generate venture hypotheses is what I'll call the biology first approach. So in this approach, you're trying to look for new areas of biology, so a new disease-associated target, a better understanding of a biological process or pathway that's implicated in disease, or maybe a new disease model. And then you identify and validate specific targets within that area of biology, and then just run drug discovery campaigns against those targets. So this is perhaps the most common phenotype of therapeutic startup. And to succeed, you need clear and well-understood biology, a translational plan that's clear, the target needs to be well-validated, and you need to execute a good drug discovery campaign. Because once a target does become validated and interesting, it develops a lot of competition. There's a lot of companies that will have great competency in executing drug discovery campaigns and can execute campaigns against novel targets once they decide they're interesting. So that's one approach. A variant of that biology first approach is what I'll call better version of X. So the prior approach, the biology first approach, you're running discovery campaigns against new targets or new areas of biology. And in this approach, you're developing better drugs against known biology. So these better drugs can be enabled by a more nuanced understanding of a pathway or target, maybe a better defined patient population that's likely to respond, or maybe there's a new technology that allows you to interact with that pathway in a way that you couldn't previously. So one example of this approach that I like is Delinia's. So Delinia developed a better version of IL-2, which is a cytokine, to treat autoimmune disease. So there was a decent amount of literature supporting the use of low-dose IL-2 therapy to treat several autoimmune diseases. So there were clinical studies that showed you dose low-dose IL-2, you can improve outcomes in these diseases. However, the immune modulating activity of IL-2 is very sensitive to dose. If you dose too low, you don't get the tolerance-inducing activity you want. If you dose too high, you actually stimulate the immune system rather than inducing tolerance, which is the opposite of what you want for an autoimmune disease. So Delinia's product was based on some work showing that different IL-2 receptors were responsible for the different functions of the IL-2 molecule's activity. So one IL-2 receptor was expressed on T regulatory cells, and one IL-2 receptor, a different receptor, was expressed broadly, or is expressed broadly on the immune system. So Delinia took advantage of this understanding of IL-2 biology and engineered an IL-2 mutation that potently activated the IL-2 receptor that is on Tregs while sparing the IL-2 receptor that is expressed more broadly on the immune system. And thus they were able to specifically induce the tolerance, specifically activate the tolerance-inducing activity of IL-2 without inducing the immune-stimulating action of IL-2. So in short, they took an existing yet imperfect solution, and then they engineered a materially better product. And for a much better overview of this particular approach, which is actually a really nice example of an investment thesis in biotech, check out an article called Rebalancing Immunity via Regulatory T-Cell Potentiation. It's an article by Michael Gladstone, who's an investor at Atlas Venture, which is a leading biotech fund, and they invested in Delinea. Okay, so those are a couple biology-first approaches. But startups don't have to be based on breakthrough cell biology. It can also be based on new technology that enables modulation of well-understood but previously intractable biology. Perhaps the canonical example of this is Genentech. So Genentech's first products were recombinant versions of proteins that had been used for decades to treat disease. Their first product was insulin, which had for decades been derived from pigs and then extracted and purified and used to treat patients with diabetes. But Genentech used new recombinant DNA technology to express human insulin in E. coli, and thus create a product more cheaply and with potential safety benefits as well. A more modern example is Avexis, for basically one of the first companies to develop a commercially viable product with gene therapy technology. I'll do a full case study on another video on Avexis because it's actually a very interesting example of finding the killer application of a breakthrough technology. But in short, there are a set of diseases, rare monogenic diseases, where we pretty clearly understand the relationship between genotype and phenotype, but we're not able to intervene in that biology with existing therapeutic technology. So specifically, if you have a disease that involves loss of function of an intracellular protein, you can't really remedy that with existing small molecule or large molecule technology. So you cannot necessarily easily deliver proteins into cells, and small molecule therapy is not clear how you're going to increase the levels of an intracellular protein with a small molecule. So gene therapy is a perfect solution to that, and that it's designed to express a specific protein within a specific kind of cell. And Avexis is a good example of building a big company based off of this novel technology. So another approach is starting with an unmet clinical need and then looking for the best way to treat that disease. So this approach is agnostic towards any particular biology or technology. You're just trying to find the best way to help patients with a specific disease. A really good example of this is a company called Prevail Therapeutics. So they're developing gene therapies for genetically defined neurodegenerative disease with an initial focus on Parkinson's. And this company was founded by Jonathan Silverstein, who's a senior partner at OrbiMed, which is the largest biotech fund in the world, after Jonathan was diagnosed himself with Parkinson's. And many companies do have this indication focus as a core part of their strategy. Even if a company is founded around core biology or technology innovations, there's often a desire to address a specific disease that's inherent in the ideation process. But it's only when you find a technology that can help you deliver a treatment that you start a company. So one last technique that used to be more popular than it is now is a build to buy approach. So in this approach, you're not focusing on any particular technology or area of biology or even an unmet need. You're really focusing on pharma as your end customer. And so specifically, you're finding a hole in a big pharma company's portfolio or an area of interest to a big pharma company, and you're delivering them a product to fill that hole in their portfolio or to address that strategic priority. So in a lot of cases, you have to have a really close relationship to the pharma company to pull this off. So a lot of cases, even with Michael Gilman's first company, he was an executive at Biogen, a big pharma company, and left to start a company around an asset that he licensed from the big pharma company. So you find assets that you know the big pharma company wants, but is too risky for them to develop. So this can be a challenging approach if you don't have good relationships within a pharma company. It's hard to keep up with constantly shifting big pharma R&D priorities, and just getting a deal done can take a lot of time and a lot of political maneuvering. And if you don't have great relationships already established, it's tough to pull this off. So the idea generation process does not have to be overly formal or structured or anything. I mean, you can really just talk to your friends about interesting science. Some investors have weekly journal clubs that are very similar to the journal clubs you may be used to in academia. If you want to focus your journal club on startups, you can just limit your discussion to articles where you can clearly articulate a potential clinical application. Include in your discussion a lot of talk about the risk profile of the idea and the ultimate clinical problem that it could address. And then another interesting way to focus your startup focused journal club is to only talk about ideas where someone is willing to reach out to the lead author or the PI to discuss the idea. So this is something that a couple of venture funds I know do, and this is just a good way to keep you moving forward and keep you goal oriented, and make sure that you're not just kind of idly talking about interesting science. All right, so that's a quick discussion of how you can generate ideas. What about triaging ideas? How do you quickly determine which ideas you should focus on diligencing more deeply? So you want to make sure that you spend some time ranking ideas to increase the chances you're investing your time the best way. With experience, you'll be able to quickly assess the potential of various ideas. But if you don't have a lot of experience evaluating, what makes a good biotech startup idea, one framework you can think about is just evaluate the risk reward of the idea, right? So basically, if the risk-adjusted reward of investing in a company is greater than that of other investments that an investor is looking at, then it's a good investment. So basically, like, there's an opportunity cost of an investor's money, and they want to deploy their capital towards ideas that have the most potential risk-adjusted reward. So the next couple slides, I'll talk about in a little bit more detail how to think about risk in biotech. It may seem a little bit academic at times or a little bit overly detailed, but I'm really just trying to provide a somewhat more quantitative framework for thinking about risk. Overall, you don't want to get too hung up in screening ideas or doing sort of like an in-depth, overly complicated exercise. You really just want to pick an idea and then get out there and talk to experts to validate that idea. But it can be helpful to have a little bit of a more structured understanding of risk if you don't already have a lot of sort of experience or intuition around evaluating ideas. All right, so quickly, the risk-reward framework is what we're going to talk about here. So first, we'll start with the reward side of things. So basically, how much money would you make, in sort of blunt terms, if the company is successful, right? How much would you return to investors if your company is successful? Very quickly, very simply, the reward in financial terms is your profit, essentially. So how much revenue do you make minus how much do you spend to generate that revenue, and then basically those are your profits. Breaking it down a little bit further, the revenue is composed of the price per patient times the number of patients that receive the product. And then the price, it can be decomposed into basically a function of clinical benefit that your drug provides. And clinical benefit is composed of the unmet medical need out there, so how sick are the patients you're treating, plus the treatment's effectiveness. How effective is your drug at improving patients' outcomes? So in general, the price you charge for a drug is correlated to the clinical benefit it provides. So a life-saving cancer drug is going to be more valuable than another treatment for the common cold. Many innovative therapeutics companies focus on areas where you can charge a high price and treat a small number of patients, rather than initially developing products that have a lower price and treat a larger number of patients. So this is similar to what Tesla did, if you're familiar with that company. It may be a little bit easier to intuit around a company like that. So they started with a higher priced, lower volume car, the Roadster, and then moved to cheaper, higher volume products. So there was a lot of risk in what Tesla was doing and just developing cars that are engineered to run on a completely different fuel source. So they wanted to address that risk of developing high performant electric cars with sufficient range before they layered on the additional risk of scaling that manufacturing process up to a mass market level. So if you see what happened to Tesla a couple years ago, you see how difficult it is to create a low cost, high volume product. And you don't want to have your first product be one where you're taking on manufacturing and scale up risk as well as innovation technology risk. It's an important topic, and it's difficult to address it in a sort of a quick presentation, but I'll go into more depth on this topic of how to balance the trade-off between risk, price, and volume in another case study on AVEXIS. So that's a little bit about reward. So basically, again, reward is how much you're going to charge per patient, how many patients are going to get the drug, minus the costs of marketing and making the product. In addition to the reward, you need to think about the risk. So the reward is what you get in a good scenario. The risk is how likely it is that you achieve that good scenario in reality. So there's a couple points that are important here. So risk is not just the probability of technical success. It isn't just how likely is a drug to get approved, to be commercially successful, or succeed in phase 2. It's also important to think about the dollar-weighted risk. So how much money and time does it take to turn over the card to address a particular risk? So this feeds into this next concept we'll talk about, which is the difference between the total level of risk and the distribution of risk. There's a couple of really great articles by Michael Gilman that illustrate this concept pretty clearly. So these are very quick and easy reads. I recommend that you check them out. It's probably better that you go and read these articles now. It's probably better use of your time than listening to the rest of the presentation. But if you want a quick summary of them, you can listen on. But I really do encourage you to read these articles. So in short, Michael is identifying the trade-off between, or not the trade-off, but he's identifying the difference between the total level of risk and how that risk is distributed in a drug development program. So we'll talk about that more on the next slide, but to quickly bring that point home, let's consider two hypothetical programs, and you can think to yourself which one you prefer. So program 1 is an antisense oligonucleotide administered intrathecally to babies to promote an alternative splice in a pseudogene to replenish a protein missing by mutation. So basically, there's a genetic disease that affects babies where they don't have an essential protein, and you're delivering oligonucleotide therapy to replace that protein, essentially. Second program, a small molecule inhibitor of an aminokinase for rheumatoid arthritis. So as of 2014, when the post was written, program 1 was certainly the riskier program. Antisense oligonucleotides were a fairly nascent modality, and the biology of using this alternative splice to increase the levels of missing protein was somewhat unproven. On the other hand, for program 2, there were several approved aminokinase inhibitors for arthritis. So the risk of the drug working in program 2 was pretty low. There was some risk, however, on the commercial side because there was a lot of competition. So you don't really know whether your particular aminokinase inhibitor is going to be competitive on the market. So which one was more compelling to you? So Michael's argument is that while program 1 is riskier on an absolute basis, it's more attractive than program 2 because of the distribution of risk. So what does that mean? So in program 1, you can spend just a few $1 million and remove nearly all the risk in the program. Once you know that your oligonucleotide increases the levels of the missing protein, just based on the understanding of the biology of the disease, you can be fairly confident that the drug is going to improve outcomes for patients. And because it is a very severe disease with no available treatments, if you have something that works in the clinic, it's likely to do well commercially. In the case of program 2, the main risk is commercial. And you can't address the commercial risk until you complete large phase 2 and 3 studies to elucidate the clinical profile of the drug and see if it stacks up well to the competition. So you can remove most of the risk in program 1 on a few $1,000,000, but for program 2, you're going to have to spend hundreds of millions of dollars before you can meaningfully reduce the risk. So there's a couple interesting corollaries to this. Firstly, in other industries, you create value by growing revenue, profits, or users. But in biotech, you create value by eliminating risk. So if you can eliminate a lot of risk early on in your program, you create a lot of value early on. Obviously, this is very attractive to early stage investors, and a lot of early stage investors and entrepreneurs focus very heavily on how they can create value and reduce risk as early and cheaply as possible. So the author of these articles, Michael Gilman, mentions a couple other corollaries. The first is that the first experiments you do that bear essentially all of the risk in the program should be as cost effective as possible. So you should get the most bang for your buck in terms of reducing risk per dollar spent. The second corollary is that the largest checks should bear little residual risk. So by the time you're underwriting big clinical studies, the risk of the program should be fairly low. So you should think about the distribution of risk even before you pick an idea to work on. It's a very important and useful way to screen your initial ideas. And once you do market research, you'll have a better idea of where the major risk points are and what it would take to remove those risks. But it's helpful to think about it up front. So this chart is intended to help you deepen your intuition about the distribution of risk in drug development. So where in the drug development process is the risk the greatest? You may have seen this chart before. It's from an older paper, somewhat controversial in its methods, but is probably the most well-cited and influential paper on the cost of drug development. It breaks down the cost of drug development into 3 components: the actual dollars spent, the cost of failure, so how many failed programs does it take to get one successful one, and then the third component of cost is the cost of capital. So you need to raise money to fund these programs, and to attract investment into a program, you need to promise investors a return. So that financial return is your cost of capital, basically. So what this chart does is it breaks down the drug development process into specific discrete stages. And then, based on data from a lot of large pharma companies, it calculates the probability of technical success at each stage. So how likely is a program to advance successfully through each phase? How much does it cost to complete each phase? And then what is the cycle time for each phase? How long does it take to get through lead optimization or phase 2 studies? And then with that data, they calculate the number of programs that you need active at a particular time to get one approved drug. So if you're in the hit to lead stage, you'll need to have 19.4 programs in the hit to lead stage if you want to end up with one approved product. So this is a sort of a detailed and slightly technical paper or chart, but it is an important and helpful way to quantify the understanding of risk in drug development. What is perhaps more instructive is analyzing which factors are the biggest drivers of the cost of drug development. So what really moves the needle? And according to this analysis, the 2 biggest drivers of the cost and risk of drug development are the probability of success in phase 2 and the probability of success in phase 3. So if you're not familiar with the drug development process, basically phase 2 is the first time you test whether a drug is preliminary effective in humans, and phase 3 are your larger and definitive safety and effectiveness studies. Up until that point of phase 2 and 3 studies, you haven't really tested the hypothesis of whether your drug is effective at treating a disease in humans. And essentially this chart suggests that the biggest reason that developing drugs is difficult is because we're not good at predicting which drugs will work in phase 2 and 3. Tying that back to the articles from Michael Gilman, essentially what he said is that risky phase 2 and 3 studies push out the distribution of risk, and programs that decrease the risk of phase 2 and 3 failure by failing early or failing fast are more attractive on balance. So the takeaway here is front load risk. More specifically, if you can reduce the cost and time to getting to human proof of concept, or having a program where there's more technical risk than clinical risk, then that will, on balance, be more attractive than a program that has maybe less technical risk, but has a larger amount of risk later on in the development process. So this venture exploration project itself is a way essentially to front load risk. The goal of this process is to subject your ideas to negative feedback so you can identify and address as many risks as possible without spending any money or any time or quitting your job to start a company. So how do you actually front load risk in practice? One way is to systematically evaluate the different types of risk inherent in the program, and then evaluate the magnitude of each risk and the cost and time required to reduce these risks. So Michael Gilman pointed out 3 types of risk: biological, clinical, and commercial. At the risk of overcomplicating things, we can add some additional criteria to that list. So we can call it additional types of risk, and then also include some attributes to measure the potential reward. So these are just an example of things you can think about when you're evaluating different ideas. It's by no means an exhaustive or definitive list. It's just a way that can get you started. And again, you don't want to overly intellectualize or make this process more complex than it is. The goal here is to really take all of the ideas you have, figure out which ones are the most interesting, and then as soon as you can, pick one idea and dive into it. There's only so much you can know before you get out and talk to experts. So again, this is just a tool to help you think through and conceptualize the level of risk in your ideas. So the next few slides, I'll just go through these specific things in a little bit more detail. The first one is severity of unmet need. Basically, this is how sick are patients, how big of a problem are you dealing with. On balance, companies or ideas where you're helping patients who are more sick or with life-threatening illnesses are on balance going to be more attractive than developing a treatment that isn't going to be as impactful to patients. Another attribute is the degree of potential clinical benefit you can provide. So how much can you help patients? It isn't just about how sick patients are. You actually have to deliver a lot of value to patients to have a viable product. Ideally, you're adding multiple years of healthy, high-quality life to patients. The most attractive drug development programs actually literally do save lives. It doesn't have to hit that bar to be an attractive program, but it's so hard and risky to develop drugs that unless you can save someone's life, it's very, very difficult to get a commercially and financially viable product. Another thing that Michael talked about was biology risk. Ideally, quickly, biology risk is basically how likely is your technology to modulate the biology in the way you think you will. So I think Michael Gilman puts it in this way. If you push a particular button in a pathway, will that pathway or biological process respond in the desired way? Depending on the program, you can demonstrate this or address this risk in vitro or in animal models, but sometimes you have to get into humans to really understand whether you're modulating biology the way you want to. Ideally, again, you want to have a well-understood target, pathway, you want to understand the biology of the disease, and you want to be able to measure at all stages of the development process how your drug is impacting the biology that you're looking to modulate. One other major aspect of risk, clinical risk. So basically, if you can modulate the biology how you want to, how likely is that to translate into clinical benefit? So how clear is the link between your target and your pathway and outcomes that matter to patients? Related to that, how long will it take to demonstrate and verify and test your clinical hypothesis? Are these going to require long and expensive studies, or can you design a nice, quick, objective study that can give you a lot of information about the clinical usefulness of your product? Ideally, you want to even have a good idea a priori that this thing is going to work in the clinic. So if in some cases you actually have evidence from randomized controlled clinical studies that drugs targeting the pathway improve outcomes. So Delinea is sort of an example of this. You know from the literature and clinical experience that low-dose IL-2 is an effective treatment for autoimmune disease. That is a pretty attractive clinical risk profile, and that's one of the reasons that Delinea was acquired when it was preclinical, before even being tested in humans. Another attribute is technical risk. So how likely is it that you can develop the drug that you want to develop? how likely is it that you can develop a drug with the acceptable characteristics that you would need to have for it to be a viable commercial product? Ideally, you want to be able to develop this with existing technology that works pretty well. But if you can't do that, that's not a deal breaker. Technical risk in some cases can be assessed pretty early on in the development process. Another attribute, competitive risk. So ideally, you want to have potential for to be first in class or best in class product. So you either want to be the first product of a particular type of drug on the market, or you want to be better than other drugs. It can be difficult if you are not in one of these categories. So follow-on or quote-unquote me-too products are increasingly difficult to develop in an environment which is much more price sensitive. There's obviously a lot of talk about price regulation in the public sphere in the United States, but there is even before any government action that may or may not happen, private payers have become increasingly sensitive to price in recent years. So another attribute we already talked about a little bit is the distribution of the risk. So really this is sort of a way to aggregate and synthesize all of the other risk attributes of a program. And again, ideally, you want to be able to take out as much risk as possible early on in the program before large clinical studies. So a lot of the prior attributes discuss the overall level of risk, but it's very important, perhaps more so, to think about where that risk occurs in your program. A couple more. So availability of IP is obviously an issue. If you're infringing on someone's patent to develop your product, that's probably a non-starter. In an ideal situation, you want to actually be able to establish a leading IP position of your own. And then last but not least, you want the idea to be a fit with the team. You don't necessarily need to have world-leading expertise on the founding team, although it helps to build that very quickly into either a scientific advisory board or the core team. And you don't necessarily need your CEO to be someone who's a 30-year veteran of Genentech, but drug discovery is a very complex and technical multidisciplinary process. So if you can bring in experienced operators, the earlier you can do that, the better. You don't have to be CEO, but on the management team. And you also want to be excited about your idea. If you've thought of a bunch of ideas and you've talked to people, done research, and found something that seems really, really compelling and attractive from a financial and clinical perspective, but you're just not really passionate about it, it's probably not worth pursuing, because even the best ideas are going to encounter a lot of difficulties and ups and downs in the process. And if you're not passionate about and interested in your idea, it becomes harder to have the tenacity to stick with a program through the ups and downs. Again, finally, you want to talk to people. This is really the whole point of the program, is to get out and talk to people and solicit critical feedback as quickly as possible. I talked a lot about some sort of theoretical frameworks for coming up with and screening ideas in this presentation. If you don't need those, then don't use them. It's really just to help you understand a little bit more what makes an interesting idea. But really, you can't know in advance whether an idea is good or not. You really have to go out and talk to people to determine whether your idea is promising. So don't get caught up in this stage. You don't even need to spend any time on this at all if you have ideas that you've already thought of that are interesting. The best thing to do is to go out there, talk to people, and get started. But if for some reason you're having difficulty doing that, then maybe some of these frameworks can help you get going.